and then went on to do master's degree in mechanical engineering, big jump, in UCSD, and then came back here to do biophysics PhD program. So he was courageous, commendable, changing different fields. He joined Michigan in 2011 in the fall semester. Subsequently, he joined my laboratory to do research on amyloid protein aggregation and structural studies. So he fully focused on mainly on Abeta, Alzheimer's disease Abeta. I will tell you the story about it. It's a very challenging project. So I charged him because he was so focused, so enthusiastic. I said, Sam, go and get the football from this amyloid field, because the football player is the running back. So we made him run forward and get the football. Okay? So there were a lot of ups and downs, but Sam really, made through the discovery of Abeta oligomers, which is the main outcome of this thesis research. Is really, really important contribution to the field and to my lab. So Sam has made outstanding contributions to my lab. In addition to research, he was safety officer. He was the go-to person for everybody to get trained and get blessings as well. He was a great mentor for a lot of undergrad students and beginning grad students. He was involved in many aspects of running the lab, including the financial aspects, supplies, and so on. Sam did use NMR, sophisticated NMR techniques, including solid-state NMR spectroscopy, and measured the coherent dipole-dipole interaction. He'll tell you later on what it is. Okay? So this coherent dipole-dipole interaction enabled him to assemble this, systematically assemble this monomer. So the so-called systematically assembled monomers, SAMs, called SAMs. Sam discovered SAMs. That's the oligomer. So today's story is going to be about SAMs by Sam.
Wow, thank you, Rams, for the wonderful introduction. He keeps insisting on calling them systematically assembled. I said randomly assembled monomers, i.e. Rams. So today I get to tell you a little bit about that. There's some other newer stuff that I worked on as well, which is exciting as a scientist. As scientists, we get one of the most exciting things we get to do is tell others about our research. The most exciting thing, of course, is good data. So hopefully I've assembled some good data here for you to listen to, particularly in the area of, as Rams pointed out, amyloid beta misfolding as it pertains to Alzheimer's disease. So you'll hear a lot about these amyloids today, amyloid beta in particular, and that's because they're associated with a number of diseases, Alzheimer's being one of the most prevalent, of course. And the common feature of all of these amyloid proteins is that they aggregate to form these amyloid plaques. The common feature of these amyloid plaques is that they form these long, unbranched fibrous structures known as amyloid, or the amyloidogenic state of these proteins. And they're not just associated. Here's 5 examples of brain diseases, but as listed here, there's 40 or so associated amyloid diseases now. And they're not just in the brain, they're all over the body. Type 2 diabetes, there's amyloid light chain, which occurs in the heart, so highly prevalent in a number of diseases. Alzheimer's is the behemoth among all of the amyloid diseases. It affects 5 million people in the US. There's a couple other scary statistics here. The scariest in my mind is that only 5% of cases happen to those people who are genetically predisposed. So that means anybody really can contract this disease, and it is fatal. And there's no cure. So just to scare you with that last final bit of information. And what happens is you get this loss of brain mass, and the question is why. Really, we don't have an answer to why right now, but Abeta is believed to be implicated in the disease. So that gave rise to the amyloid hypothesis, or the amyloid cascade hypothesis, in this case particularly for Alzheimer's disease. And this schematic here, you'll see a lot of these schematics about amyloid aggregation here. And this really is the problem of amyloids is that they start off in this monomeric state and they end up in this fibrillar state. Now it's relatively well accepted that both of these states are relatively benign. And it's these intermediate states that lead to the highest degree of toxicity. And they elicit cellular toxicity. You inject them into mice, and mice get Alzheimer's-like symptoms. So there's a lot of prevalence for these being implicated in Alzheimer's disease. Of course, there are a number of theories as to how they elicit toxicity, kind of some of the examples listed here. But the general feature of all of these problems is neuronal dysfunction and neuronal cell death. In our lab, we focus on this end of the spectrum in the membrane disruption. It's really, from a biophysical perspective, biophysics is kind of well armed to study this end of the spectrum here. And my early work in the lab focused on these two forms of membrane disruption here, and I'm just going to summarize that work because it kind of laid the groundwork for everything else.